Hey guys, welcome to topic 3 in chapter 8. This topic is going to contain a lot of new information just like topic 2 did, and so it's really important that you understand how these post-transcriptional controls we're going to talk about in topic 3 work. This is going to be a shorter lecture, so if you feel like we went through things too quickly or if something's still fuzzy as I fully expect it to be, please let me know. We only have one objective for this topic, and it's just to explain how post-transcriptional controls work and what they are. And so make sure you understand what those are, and as I said, if you're not sure, make sure that you bring it up in class so that we can talk about it. All right, so let's start here. Remember this picture from the last topic? Last topic we talked a lot about number 1. This time we're going to talk a lot about number 5. So it's important that you see how that works, and number 4 to an extent as well. There are number 2 and number 3 aspects of it, so don't blow those off. And as I said, this image is the hallmark of chapter 8. If you can understand and discuss this image and examples, you're going to be golden for the chapter 8 section of the exam. So there's a couple controls we're going to talk about within here. We're going to talk about ribose switches, miRNAs, and RNAi within this whole process of post-transcriptional controls. And most of my examples are going to be prokaryotic, and that's just simply because we've worked them out in prokaryotes. We understand how they work because they're just simpler organisms. That doesn't mean that there aren't some of these in eukaryotes. It just means that either we haven't found them or we do know about some of them, and some of them we've mentioned, or I will mention rather. But these examples are all prokaryotic examples here. And so you can see that we have 4 main post-transcriptional controls in mRNA that the cell will use. And the first one of these is just a simple repressor, just like we talked about with the tryptophan operon in the last topic. A repressor will sit on that start codon and prevent the ribosome from binding to it and transcribing it. The next one has a temperature increase. What happens when temperature changes from an optimal to a suboptimal temperature, the mRNA will actually change shape. And what this does is it prevents the ribosome from once again binding to the DNA or to the RNA, and then transcription can't happen, or translation can't happen rather. Another example is a ribose switch, and that's what you see here on the bottom left. And we're going to talk about this in more detail in the next one. But basically what this is there's a conformational change within the mRNA that allows for the ribosome to bind if the repressor molecule or whatever it is not there. And I'll show you that in the next one. And then the last one here is an example of mRNA. And we're going to miRNA or siRNA, there's a lot of different names for it, but microRNAs. And what it does is it binds directly to that start codon, once again, prevents the ribosome from binding. And we're going to talk about each of these, or these last 2 here for a little bit more time. So the first of these are those ribose switches. What happens is there's this extra segment of RNA that's bound to the mRNA. And when there is a small molecule bound, so in this example, guanine, when it's bound to this sequence, the polymer or the ribosome can't bind, or in this case, in this picture, the polymerase won't bind to make more of the gene. And what this does is it inhibits the translation of the mRNA into protein, because we don't need any more guanine. We have plenty of guanine here. When that guanine that's attached, you can see on this image, is used up and removed from the situation, the conformation will change, and we'll go ahead and make some more mRNA and then more guanine out of it. So make sure you understand how that ribose switch works. miRNAs are microRNAs. Sometimes you'll hear them called siRNAs for silencing RNAs, and that's more of a lab technique tool that we've used. What it is it's this matching complementary sequence of RNA that will bind directly to the mRNA. And this will make it so that the mRNA is degraded. So it's either reduced or completely degraded. So it's just a similar complex that'll do that. And there's this whole complex that works with it known as the RISC protein. You can see that here in this image. It stands for RNA-induced silencing complex. And what it does is it binds with that miRNA, finds the target DNA or the target mRNA, and breaks it down or inhibits that translation. So that's how miRNAs work. RNAis are RNA interferences, and they're just like miRNAs, except for they're not natural to the cell necessarily. We will introduce them to the cell. These are those siRNAs I was talking about. We use them in research to help degrade them, and this is an example of my research, is that I use this to degrade the mRNA product so that the proteins I want to be knocked down in a cell are no longer produced. So this is just an example of how we can utilize this tool in the lab. So as we wrap this up, this is our last slide. I told you this one was a quick topic, and as I said, if any of these are too muddy for you still, please let me know so we can work on them. But I want you to understand that one more eukaryotic post-transcriptional control, and this is the ribosome binding complex, where the mRNA is seated in a small subunit. And the repressor can bind to this untranslated region at the head of the sequence, and it prevents the ribosome from binding, and it can't find that AUG sequence. And so this is just another way that eukaryotes can prevent this. So make sure you review these items and let me know if you have any questions, and we'll talk about it in class. Thanks. Bye.